a series of intercellular membranes that compartmentalize the cell. So they are within the cell, they are intracellular, which means inside cellular, the cell, and they break the cell down into smaller sections. These membranes restrict enzymatic reactions to specific places within the cell. The endomembrane consists of the nuclear envelope, which, remember, keeps the nucleus separated from the rest of the cell. It also contains the membranes of the endoplasmic reticulum, which is often abbreviated the ER. It also consists of the Golgi apparatus, which takes things from the endoplasmic reticulum and processes them, and then ships them out in the form of vesicles. Vesicles are basically like boxes made out of cell membrane that the stuff that the Golgi apparatus made, that it processed, it's the goodies inside the box that are being shipped throughout the cell or to other cells. So there are two types of endoplasmic reticulum. Generally, they are a system of membrane channels and saccules, which are flattened vesicles, which are made out of cell membrane. They are continuous with the outer membrane of the nuclear envelope. So they start where the nuclear envelope ends. Most of the endoplasmic reticulum, not a small amount, but a lot of it is studded with ribosomes. It is called the rough ER, rough endoplasmic reticulum, because it looks bumpy whenever you look at it under an electron microscope. The ribosomes produce proteins, and those proteins are used in the ER to make other things. So the ER takes those proteins made by those ribosomes and adds sugars, which results in glycoproteins, which are important in cellular functions. These substances are then shipped to the Golgi apparatus. It's wrapped up in a vesicle, wrapped up in a box of cell membrane phospholipids, and taken to the Golgi apparatus. The smooth ER is much like the rough ER, except it has no ribosomes. And here, lipids are made. So testosterone, estrogen, those kinds of things are made by the smooth ER. Detoxification happens here. Cells that do a lot of cleaning up blood, your liver, your kidneys, they have a lot of smooth ER. Especially your liver, which detoxifies drugs. So if you're taking large amounts of ibuprofen or Tylenol, your liver is what is actually breaking that down. Once it is done making those lipids are wrapped up in a vesicle, remember the cell membrane, and shipped to the Golgi apparatus as well. Here's what the ER looks like. Here we have the rough endoplasmic reticulum. You can see that it is rough because it has ribosomes stuck to it. And as we move out from the nucleus, we have smooth endoplasmic reticulum with no ribosomes stuck to it. The Golgi apparatus, named for Camillo Golgi, consists of flattened, curved saccules that look kind of like pancakes, but they're hollow. It takes things from the ER and modifies them. So it gets them from the endoplasmic reticulum on the cis, or the inner face. It takes those materials, modifies them, and prepares them to be shipped out on the trans, or the outer face of the organelle. Some of those vesicles are transported within the cell, and some of them are secreted from the cell in a process called exocytosis. All right, sometimes those vesicles return to the ER, or sometimes they just merge with the plasma membrane. Depends on what is in the vesicle. Here we have the Golgi apparatus. The cis face is the face that faces the endoplasmic reticulum, and the trans face faces the outside of the cell. Here you can see some of the vesicles that are being produced, and this one is probably some kind of waste or some kind of substance that needs to be transported to the outside of the cell. It fuses with the cell membrane, and the inside stuff, the materials, are expelled from the cell. Here's an actual image created using an electron microscope of what the Golgi apparatus actually looks like. Life lysosomes are membrane-bound vesicles that are not found in plants. They're made by the Golgi apparatus, and they contain hydrolytic digestive enzymes, kind of like stomach acid. And they digest large molecules into simpler subunits. They're like the recycling center of the cell. In white blood cells, the digestive enzymes are what engulf the pathogen. They surround the bad thing and break it down. Sometimes lysosomes don't work properly. This is caused by a defect in some of the enzymes contained in them, and it can lead to a disease called Tay-Sachs, which is a debilitating disease that affects young children. Generally, they don't live to be more than 5 or 6 years old. It's not a good disease. If you're interested in it, you can look it up, but we're not going to spend a lot of time talking about it. Sometimes gene therapy can replace the missing enzyme in the cells, where scientists will actually take a healthy gene, a gene that is correct, and insert it into cells of the person that is affected with the disease. Sometimes it works, sometimes it doesn't. Here we have the structure of a lysosome. All right, here's a mitochondria that it is going to break down. This mitochondria is no longer functional, so the lysosome has engulfed it, and it is going to actually digest it and break it down into smaller pieces so that it can be used again, possibly to make a new mitochondria or to make something different. Endomembrane summary. Proteins produced in the rough ER and lipids from the smooth ER are carried in vesicles to the Golgi apparatus. The Golgi apparatus or Golgi body modifies these products and sorts and packages them into vesicles, and then they are then shipped out to different places within the cell or without the cell, outside of the cell. Secretory vesicles carry products to the membrane where exocytosis produces secretions and expels those substances out of the cell. So this would be like mammary glands producing milk. So the cell creates the milk, and it needs to leave the cell so that it can get to the baby that needs it. And so it is expelled from the cell into milk ducts, and then it can eventually be consumed by the baby. The pancreas produces digestive enzymes that helps break down your food. So those substances, those enzymes, are actually made inside a cell and then secreted so that it can get into your digestive system. Lysosomes fuse with incoming vesicles and digest macromolecules. So lysosomes are very important in the process of digestion. And here we have a diagram showing all of the parts. Here we have the rough ER, the smooth ER, the vesicles going to the Golgi body or the Golgi apparatus, and then the vesicles being expelled. And that's it for this video, guys. If you have questions, let me know.